Welcome back to Legalese 123, where we are covering cases across America. Hello and welcome back to Legalese 123. I'm your host, Missy K. We are back in action at the trial of Brian Walshe, day 7. So we're going to do these in segments. I thought maybe if we break it down by people who testify instead of a long trial, that maybe people might appreciate that a little bit better. So we're going to do it in segments. So we just finished up with the crime scene tech, Mr. Sheehan. We are now moving on. So let's bring this up here and let's see who we have next.
Jurors, in light of our late start today, I'm going to keep on. Everybody good? Thumbs up. You may call your next witness.
All right, we are on witness 31.
Commonwealth will call Bryce Raymond.
Bryce, someone another.
Do you solemnly swear or affirm the testimony you'll give in the cause now and hearing will be the truth, the whole truth, and nothing but the truth, so help you God? Thank you,
He
sir.
has a small folder, so let's see what that is. Usually folders mean they're going to testify to something interesting.
Thank you very much, Your Honor.
Uh-oh, breathing in the microphone.
Good morning.
Could you please state your name for the record and spell your last name?
Bryce Raymond, Raymond.
Could you please tell?
Bryce, Bryce.
Oh,
Could you please tell us your educational background?
I have a Bachelor's of Science in Forensic Science from Penn State University and a Master's of Science in Biology from Tufts University.
And what is your occupation, sir?
I'm currently a Forensic Scientist 2 in the DNA unit at the Massachusetts State Police Crime Lab.
okay, we're in the
And how
DNA
long have
unit.
you been employed at the Massachusetts State Police Crime Lab?
Since June of 2019.
And can you please tell the jury your present duties in the DNA unit and the nature of the work that you do?
Yes, as a Forensic Scientist 2, I am in charge with processing evidentiary items through our DNA lab processing, as well as interpreting and making any conclusions to known standards.
Nice.
Do you have any specialized training to be a DNA analyst?
Yes, I received at least 6 months of specialized training in which I read all the protocols and procedures at the lab. I attended lectures and took quizzes, as well as worked on mock evidentiary items through the lab processing. And at the end of testing, I take a competency test.
Nice.
And did you pass that competency test?
Yes, I did.
So does your testing and training stop after that initial 6-month period?
No, every year we have to do at least 8 hours of continuing education in order to stay knowledgeable in the field.
And do you continue to be tested at the lab?
Yes, we do.
And what kind of testing is that called?
That's called a proficiency test.
And how does proficiency testing work at the lab?
Yeah, so twice a year we have to take a proficiency test to continuously show that we have the skills and knowledge in order to perform DNA analysis. This is given by an outside accrediting body, and when we complete our work, we send in the results to that accrediting body for them to grade.
And what are the results of your proficiency testing?
I have passed every one I have taken.
Woohoo!
So approximately how many samples have you had the opportunity to analyze for DNA testing?
Approximately 2,000.
And have you testified before in Massachusetts about DNA?
Yes, I have.
Does the Massachusetts DNA unit at your lab follow standardized protocols and procedures?
Yes, we do.
And who sets those protocols and procedures?
They are based on some accrediting bodies such as the FBI, as well as validations that are done at the lab.
Well, you anticipated my next question, which is your lab, the Massachusetts State Police Crime Lab, accredited?
Yes, we are. We are accredited by ANAB, which is the ANSI National Accreditation Board, as well as the FBI Quality Assurance Standards for DNA laboratories.
And what does accredited actually mean?
That means we are following specific set of standards to make sure that the scientific work that we are performing is accurate and reliable.
And is that a national accreditation?
Yes, it is.
And does your work undergo any kind of review process?
Yes, all work goes under two different types of review processes, a technical review and an administrative review.
And is that review process required under the accreditation standard?
Yes, it is.
And so if you could just explain technical and administrative review, please.
Yes, a technical review is one that is done by a qualified analyst that takes a look at your work to make sure that you followed all the proper protocols and procedures and made sure that any conclusions you made are scientifically sound. An administrative review looks over the file to make sure there's no spelling mistakes, no grammar mistakes, all the pages are numbered and initialed, and other administrative things such as that.
And why is that all important?
That's important to make sure that we provide the best quality work that we can.
I now want to talk about DNA. What is DNA?
DNA stands for
Deoxyribose-
deoxyribonucleic acid.
acid.
It's the genetic blueprint of life and what makes you, you. You inherit your DNA from your parents, so you get half of your DNA from your mom and half of your DNA from your dad. And no two individuals have the same DNA profile except for identical twins.
Right.
Is DNA the same in every cell of a person's body?
Yes, so whether we test blood, saliva, hair, skin, we should expect the same DNA profile if they came from the same person.
So what type of biological material can DNA be found in?
So that can be blood, saliva, skin, hair, sweat, urine, anything biological.
So can you explain generally the process that is used for forensic DNA analysis?
Yes, at the lab, we have a 4-step process. The first step is extraction. This is when we break open the cells and collect the DNA. The second step is quantitation or quantification. This is when we can get an estimate of how much human DNA is present as well as how much male DNA is present. And that's so we can target an appropriate amount for the third step. And that third step is amplification. This is when we make millions and millions of copies of specific areas on the DNA and are fluorescently tagged. And then the fourth step is separation and detection, also known as capillary electrophoresis. This is when we put the DNA on an instrument that separates out the DNA by size and has a camera to detect those fluorescently tags, and that produces raw data.
And what is the goal of that raw data?
The goal of that raw data is to then be read by an interpreting analyst in order to interpret the DNA profile.
And what is a DNA profile?
A DNA profile, so the specific areas that we are copying during amplification is called STRs or short tandem repeats. That is just areas on the DNA that repeat different lengths for a person. So one person could have 5 repeats, another person could have 6. And it's the combination of all those numbers is what makes up a DNA profile.
So how many locations are you testing?
We test 26 locations as well as a sex determining location.
And what is batch testing?
Batch testing is what we do at the lab and is a common practice in forensic science, in which if many items need to be processed for the same lab step, one analyst will process all those items at once on the same instrument.
So why do you use batch testing?
We do that for efficiency, as it is better for one analyst to process, say, 50 items at one time on one instrument than 50 analysts trying to process one item each on the same instrument, one after each other.
It would take forever.
Is there a protocol that you follow prior to retrieving any evidence to prepare your lab area to avoid contamination?
Yes.
So how do you avoid contamination?
To avoid contamination, I first put on protective personal equipment, and that can include a lab coat, a mask, a hairnet, and for me, a beard net. When I go to my lab bench, I also put on gloves and I spray the lab bench with bleach, clean that off, and lay down lab bench paper before I proceed with any testing.
So why do you use bleach?
The bleach is to destroy any DNA that may be present on the lab bench.
And why do you use bench paper?
We use bench paper to prevent any contamination from items that I may be testing to be deposited onto the lab bench for the next person.
And why are you wearing PPE?
The PPE is also to prevent any DNA that I may touch to go over to another lab bench or any of my DNA to be deposited on the items.
And once your area is prepared, can you please tell us what you first did and what you first do with items that you're going to sample?
Yes, so for sample preparation, I get a list of items that have been selected for DNA analysis. I get those items out of storage in our secured walk-in room. I take that container of evidence back to the lab bench. I scan it into my custody and take a photocopy. I then open up the container, grab the items that I need, and put those aside ready for testing. I seal up the container with my initials and date and put the container away.
And did you work on lab case 23-00596?
Yes, I did.
And what did you initially do to prepare the samples for DNA analysis?
I did sample preparations on some items.
Okay, so I'm going to ask you specifically about samples. Did you prepare samples 5-1.1, sample from swabs of red-brown stained cave on basement floor? 7-10.3.1, sample from the stained area C on the blade of the hacksaw. 7-10.4.1, sample from the handle of the hacksaw. 9-22.1.1, sample from stain A on white towel number 1 with red-brown stains. 9-28.1.1, sample from stained area A on apparent hairs. 9-31.1, sample from cutting of red-brown stain from piece of rug from bag number 5 from 300 Forest Street. Did you prepare samples from those?
Yes, I did.
So after preparing the samples, what did you next do?
Six
After
samples.
preparing the samples, I then did the first lab step of extraction.
And how did you do the extraction step?
Yes, to each of the tubes along with the controls, I added chemicals to the tubes and put them in an oven. And that is done to break open the cells in order to free up the DNA. I then take those tubes and put them on an instrument that separates out the DNA and puts them in a
Centrifuge.
separate tube in liquid form, and that
Sends 'em-
tube is called an extract.
Did you use equipment?
Yes, I did.
What equipment did you use?
I used an oven, and the instrument was an Easy One robot.
And how did you use the equipment?
Easy One robot.
For the oven, I just placed the samples in and took them out. For the Easy One robot, I put the samples on the robot and then programmed the robot with the proper protocol to use for the extraction and collection, and then got the tubes off of the robot when the process was done.
And I think you said that that is now called, well, what is that now called?
That is now called an extract.
And then what did you do next?
Next, I took a small portion of the extract, known as an aliquot, and put that into a separate tube and prepared that for the second step of quantification.
So now we're on step number 2,
Yes.
okay? And what items did you do quantification for?
The same items that were previously mentioned.
And how did you perform the quantification step?
Yes, so for each of the aliquots, I put a portion onto a 96 well plate along with other chemicals. I then placed that 96 well plate onto an instrument, and that instrument does a series of heating and cooling, and that produces the data that gives us the estimation of DNA.
What's
So
the
I believe you
instrument?
just named the equipment that you used.
Yes, it is a real-time PCR system.
Okay. And so how did you use that equipment?
So I put the 96 well plate onto the instrument. I then used the computer program associated with it to say that to start the quantification process. And then after it was done, I collected the data off of the equipment and then put it on our computer drive.
And then what did you do next?
After that, I discussed the quantitation results with the interpreting analyst of this case, who is Suman Salim.
Summon
And
Salim.
then what did you do?
Then we came up with how to do amplification strategies for each of the items. And then once that was decided, I went forward with the third step of amplification.
So step number 3 is amplification.
Correct.
And what items did you perform amplification for
Those same 6 items.
that we previously named?
Correct.
Okay. And how did you perform the amplification step?
Yes. For each of those items, I got the extracts, and I either used all of the extract or a portion of the extract to target an appropriate amount of DNA for testing.
Let me just stop you for a second. What's extract?
Extract was the product from the first step of extraction.
Okay. And now finish your answer. I'm sorry to interrupt you.
Yes. So for those samples, I added chemicals and put them into an instrument known as a thermocycler. And that also does a series of heating and cooling, which makes the copies of the DNA that are fluorescently tagged. And at the end of that process, the tube is now known as an amplicon.
And then what did you do next?
Next, after the amplicons were done, I then proceeded on to the fourth step of separation and detection.
And which items did you do the fourth step for?
The same 6 items that were mentioned.
So you did all the steps of DNA for those 6 items.
That's correct.
And how did you perform the detection step?
Yes. Of the amplicons, I took a small portion of each amplicon and put it on a 96 well plate, added chemicals to the plate. I then put that plate onto an instrument known as a 3500 genetic analyzer. I then programmed the genetic analyzer to perform the separation and detection. And then the instrument separated out the DNA fragments by size and with the camera detected those fluorescent tags.
And then what happens next?
After that data is generated, I take the data off of the instrument and put it on our computer drive.
And then what happens?
After I'm done, I let the interpreting analyst know that data has been collected, and I hand over any worksheets that I generated to them.
And who does the interpretation?
In this case, it was Suman Salim.
So you did not do the interpretation here.
I did not.
I might have one moment, Your Honor.
So he does all like the sciencey stuff to get the DNA. She interprets it.
Nothing further, Your Honor.
Cross-examination.
Just a few questions. Mr. Rigg. Good morning.
Good morning.
And you've taken a lot of proficiency tests. You've passed them all, correct?
Yes.
You take pride in what you do, don't you?
Yes.
And you're running these machines, the analyzer and everything else, and you're running them consistent with the protocols that are required to be followed to run them accurately, correct?
That is correct.
And what you do is you depend on the fact that the samples that you get for testing are samples that will result in reliable results, correct?
Correct.
But what is your height at the lab?
I am a forensic scientist too.
Okay. And are there forensic scientists 3 and stuff like that? Forensic scientists 1?
Yes.
Okay, but you're a 2, correct?
Correct.
And with your knowledge and experience It's fair to say, isn't it, that you have no idea where the samples that you subjected to DNA testing came from, correct?
That's correct.
And in fact, you have no idea how they were collected, wherever they were collected from, correct?
That's correct.
And in fact, you can tell the jury that at times when you conduct testing like you just described, the results will indicate that there are mixtures of different DNA in a particular sample, correct?
That's correct.
And whenever you get the results that result in a mixture, you can't determine, as a forensic scientist 2, for that matter, forensic scientist 3, you can't tell how those two different substances came into contact with each other, resulting in a mixture, correct?
Our testing does not determine how DNA got onto an item.
And hopefully it's a simple question. Your DNA results do not tell you anything about what happened at a, let's just call it a crime scene, what happened at a particular scene or where they were collected, correct?
Correct.
Thank you, Steve.
Any redirect?
No, Your Honor.
That was
Witness may step
pretty
down.
straightforward. So, why did this witness have to testify? So, this witness testified that he did the extraction, the amplification, the quantitation, the end all be all, right? He is the one that said, I got the samples, I extracted the DNA from them. And then he turned it over to his next person who will be hopefully witness 31. Now she's getting ready to say something. Let's see what she has to say.
So jurors, with that little extra bit, we'll take our morning break. We'll be back in 20 to 30 minutes.
Okay, we're going to zoom. Only fan, the fan that is the only fan. Hopefully. All right, let's see.
Sure that they are. Could I have 1 second to check?
Oh, shoot. Let's see. We're back on the record with the lawyers.
My apologies. Do we have Mr. Mutlu? Jim? Mutlu?
No, you didn't hear me.
I'm sorry.
I asked you, do we have, maybe I shouldn't be so abbreviated with my words. Do you have a non-Mr. Mutlu witness ready?
Yes.
Let's call that witness and get the jury back in here.
Yes, Your
I've
Honor.
had a little lumpy morning and I want to continue keeping the flow.
Yes, Your Honor.
I just want to make sure they're still sitting out on the bench. I'm not sure that they are. Could I have 1 second to check?
Sure.
Okay, so Mr. Mutlu is up to be testifying, but maybe they're not ready yet because there's some controversy she still has to work out. So anybody but Mr. Mutlu. Interesting. All right, so witness number
Great.
32.
Get the jury back in. We'll do Mr. Mutlu over the break.
Yes,
And
Okay.
over that, I mean, over the lunch break.
Yes, Your Honor.
Apparently, maybe they want to voir dire him or something like that. What is he going to testify to? So let's zoom because we have the room. So let's get down. Let's get this party
Commonwealth may call the next
started.
Witness.
Thank you very much, Your Honor. The Commonwealth would call Brianna Kiesel.
Brianna. So apparently we're not going from one DNA tech to the next, which I find very jumping around in the story, but that's okay. Here we are. Oh, she's got a file folder. So what'd she do?
Do you solemnly swear or affirm the testimony you give in the cause now and hearing will be the truth, the whole truth, and nothing but the truth, so help you God? Thank you. Please seat.
All right. Brianna, what have you got for us? She got a file folder. Not very
You may
thick.
proceed.
Thank you very much, Your Honor.
She
Good
looks
morning.
nervous.
Can you please state your name for the court and spell your first and last name?
Sure. My name is Brianna Kiesel, Brianna. Last name Kiesel, Kiesel.
And what is your educational background, ma'am?
I have a bachelor's in biology from Arcadia University and a master's in forensic science from Towson University.
And what is your occupation?
Currently, I'm a forensic scientist 2 with the Massachusetts State Police Crime Lab,
And what unit are you assigned to?
the DNA unit.
And how long have you been employed at the Massachusetts State Police Crime Lab?
6 years.
And what are your responsibilities as a forensic scientist 2?
I am authorized in processing, interpreting, and reporting samples that were collected from crime scenes, as well as standards collected directly from an individual. I can also technically review other analysts' work when it concerns batch files, which we call files that have standards collected directly from an individual.
And do you have training for this position?
Yes, I do.
Can you tell me about your training?
Sure. Our training is a minimum of 6 months, and during that time, we have lectures, quizzes, and this all culminates into a final written exam. We also have a practical component in the laboratory where we take several steps for sample sets forward, and that all culminates into a final written exam, as I mentioned, and a final lab practical.
And did you pass that written exam?
Yes, I did.
Yay.
And that's called a competency evaluation?
Yes, that is a competency, the lab portion of it.
And do you also have to take proficiency testing?
Yes, we do.
And can you explain that to the jury?
A proficiency test is an ongoing competency test where we have to take samples forward twice a year to prove that we are that we are knowledgeable and still performing that skill or method according to laboratory standard operating procedures.
And have you passed your proficiency testing?
I have, yes.
And have you testified in court about DNA?
I have.
So I want to now talk to you about this case 23-00596. Prior to retrieving any evidence in 23-00596, how did you prepare to process the evidence?
So once I was assigned the samples, I cleaned my bench space with bleach and wiped everything down, every instrument that I was using. And once that was done, I laid down a piece of fresh bench paper.
And directing you now to lab number 23-00596, did you work on items 9-27.2.1, the sample from the interior of the dark gray slippers from bag 4 of 300 Forest Street, and item 9-27.3.1, stain A of the exterior left gray slipper?
Yes, I did process those samples.
And what work did you do on those 2 items for sample prep?
For sample prep, they were already prepared by our criminalistics unit. So I just went into a secure freezer area to retrieve the packaging. I removed the tubes from the packaging and just visually examined the items within the tube.
Did you also work on item 9-32.1.1, the sample from the interior sleeve cuff of the Tyvek suit 9-32.2.1, the sample from stain A on the exterior left pant leg of the Tyvek suit, and sample from stain D on the interior right sleeve of the Tyvek suit?
I did, yes.
And what work did you do for that?
Those samples I performed just like I previously mentioned, where I went to the secure freezer area, retrieved the packaging, removed the tubes from the packaging, and just observed the samples within the tube.
So you also just did the sample prep for that?
Yes.
Did you also work on item 16-13.1, the unknown tissue recovered from the Swampscott dumpster?
Yes, I did.
And what work did you do on this item?
So in this case, this item was still in a specimen jar. So I retrieved the packaging, just like I previously mentioned, made sure the seal was secure as I did with the other samples. And then after that, I opened up the jar and sampled a few items from the jar and placed it into a tube for processing.
So you actually had to make the sample for this?
I did, yes.
Okay. And how many samples did you make?
So this was 16-13.1. There were two tubes we refer to as 16-1, or excuse me, 16-13.1A and 16-13.1B.
And did you do any further work?
No, that is where my work ended.
Okay. And where did the samples then go?
I then passed the samples off to Madison Frank for extraction.
Cross-exam. The witness may step down.
Thank you.
All right, witness 32, Brianna Kissel just made samples.
Commonwealth may call its next witness.
Okay,
Commonwealth would
witness,
call Madison Frank.
witness number 33, Madison Frank. She's probably going to testify almost exactly as Mr. Bryce Raymond did, and probably going to have the same 4 things. So we'll see. I could be wrong. I'm not a scientist, but I know some information.
Do you solemnly
All
swear
right,
or affirm
science,
the testimony
science,
you'll give in the cause
science.
now and hearing will be the truth, the whole truth, and nothing but the truth, so help you God? Thank you.
She blinded me with science. If you're not singing along, all in emotion.
You may proceed.
Thank you very much, Your Honor. Can you please state your name for the court, spelling your name?
My name is Madison Frank. That is MADISON
And what is your educational background, ma'am?
I have a bachelor's of science degree in forensic and investigative sciences from West Virginia University.
And what is your occupation?
I'm a forensic scientist too in the DNA unit of the Massachusetts State Police Crime Lab.
And how long have you been employed at the Massachusetts State Police Crime Lab?
I've been employed there since October of 2019.
And what are your responsibilities?
As a forensic scientist too, I will perform DNA analysis on either known or evidentiary biological samples, interpret those results, and report out my results in a case file.
And have you had training for this position?
Yes, I have.
And have you passed your proficiency testing?
Yes, I have.
And have you passed the competency testing that was part of your training?
Yes, I have.
And have you testified in court about DNA?
Yes, I have.
Directing your attention to lab number 23-00596, did you do work on this case?
Yes, I did.
I want to ask you now about items 9-27.2.1, the sample from the interior of the dark gray slippers from bag 4 of 300 Forest Street, 9-27.3.1, the stain A on the exterior left gray slipper, 9-32.1.1, the sample from the interior sleeve cuff of the Tyvek suit, 9-32.2.1, the sample from stain A on the exterior left pant leg of the Tyvek suit, 9-32.5.1, sample from stain D on the interior right sleeve of the Tyvek suit, and 16.13.1, the unknown tissue recovered from the Swampscott dumpster. I believe I just gave you 6 of those samples. Did I mention 9.32.2.1, the sample from stain A on the exterior leg pant of the Tyvek suit? So those would be the 6. What work did you do on those?
DNA analysis is a 4-step process, so I perform steps 1 and 3 of that process on those samples.
Okay. And what are 1 and 3?
Step 1 is extraction, and step 3 is amplification.
Okay. And how did you do step 1, the extraction step?
For extraction, I received the samples and I added chemicals to the samples and the controls, and then put them in an oven so that we can break open the cells to obtain the DNA. Then those samples will go on an instrument that helps to clean up the sample so that we're only dealing with the DNA and no other cell parts. Then after the extracts come off of the instrument, then I will take a small portion of that sample and I will put it in a new tube to be used for the second step.
And which instrument did you use or equipment?
An easy one robotic instrument.
Okay. And how do you use that instrument?
The samples will go onto the instrument in tubes, and then there will be new tubes where the samples will go through processing in the instrument to put out just the liquid and DNA portions of the sample.
And then what is that biological material then referred to?
An extract.
And what is that extract then ready for?
The extract is then ready to be quantified in the second step.
So did you do that second step quantification?
No, I did not.
Who did that second step for those 6 items?
Troy Adams did the
Oh,
second
boy.
step of quantification.
And why is that?
We use a batching process when we are processing our samples so that it is a more efficient and streamlined processing.
And are you qualified to do all of the steps of DNA, the 4 steps that you described?
Yes, I am.
And then did you do more work on this case with the 6 samples?
She said yes.
Yes, I also
3.
did the third step of amplification.
On all 6 of those samples?
Yes.
And that being 9-27.2.1, 9-27.3.1, 9-32.1.1, 9-32.2.1, 9-32.5.1, and 16-13.1. Did I get that right?
Yes, that is correct.
Okay. So you did the third step. And how did you do the amplification step?
So I took the extracts that were generated from the first step, and I took portions of those samples and added chemicals to the tubes with these samples. And then I put them onto an instrument called a thermocycler. And this will have the samples go through periods of heating and cooling. And this is to make millions of copies of the specific DNA that we are looking for to be able to visualize it in further down in our processing.
And which equipment did you use for that?
A thermocycler.
Okay. And then how do you use that equipment?
The samples just go on, they are in small tubes and they will go on into small holes in the thermocycler. And then it is a like metal plate that they are in, and it will go through different steps of heating and cooling.
And what happens next to the DNA process?
So the next step will be those tubes that just went through the heating and cooling, that those tubes will be used for the 4th step of separation.
And who did that step?
Troy Adams.
So you did not do that step.
No, I did not.
And did that end your work on this case?
Yes, it did.
Let me guess. Is Troy Adams up
Any
next?
cross-examination? The witness may step down. Thank you.
Thank you.
All right. That is 33. Oh, boy. Witness number 34. Can I get a witness? I don't know the rest of the words.
Next witness.
The Commonwealth would call Troy Adams.
Yes. All right. Okay. Troy did step 2 and 4 of this. And then I want to know who analyzed it. Maybe that's why they didn't come
Do you
call
solemnly swear or affirm the testimony you'll give in the
Suman
cause now and hearing
Celine,
will be the truth, the
because
whole
maybe
truth, and
she
nothing
did
but the
all of the analysis of all of them. So we'll see. All right, Mr. Troy Adams.
You may proceed.
Thank you. Can you state your name for the court, spelling your first and last name?
My name is Troy Adams, Troyadams.
Thank you. And what is your educational background, sir?
I have a bachelor's degree from Pennsylvania State University in forensic science.
And what is your current occupation?
So I currently work as an investigator for the Lehigh County Coroner's Office in Pennsylvania.
And
Oh.
how long have you been in Pennsylvania?
I've been in Pennsylvania for approximately 3 years now, 2 years working in my current job.
So prior to moving to Pennsylvania, did you work in a different field?
No.
Did you work for forensic sciences in Massachusetts?
Yes.
And can you detail your working history as it pertains to forensic sciences?
Yes. So out of college, I joined the Massachusetts State Police crime lab there as a DNA analyst. I completed all my training protocols and was tested for competency. And then I began performing casework on samples.
And then did you work for a different company in Pennsylvania as well, doing forensic sciences?
Yes. After I left the Massachusetts State Police, I joined Quest Diagnostics as a forensic scientist in their toxicology division.
And just keep your voice up so everyone can hear you.
And have you had training for your position at the Massachusetts State Police Crime Lab and also for your position at Quest?
Yes.
And have you had ongoing training as well?
Yes.
And have you taken proficiency tests?
Yes.
And what were the results of your proficiency tests?
I passed every proficiency test.
And do you have any certifications in your current job?
Yes.
And where are those from?
So I am certified as a national medical legal death investigator.
And how did you receive that?
So I received that after a certain amount of training at my current job, and then going through and taking a certification test.
I want to direct you now to when you were working in Massachusetts. Did you do work?
Very interesting, right? So he started out as he's this DNA science dude, and now he's like, no, I don't want to do this anymore. I'm going to go into toxicology, but now I can do certified death. That's pretty cool. So get a science degree, children.
On lab number 23-00596.
May I refer to my file?
You may.
Yes.
And did you do work on items 9-27.2.1, a sample from interior of a dark gray slipper, 9-27.3.1, stain A on the exterior left gray slipper, 9-32.1.1, the sample from the interior sleeve cup of a Tyvek suit, 9-32.2.1, the sample from stain A on the exterior left pant leg of Tyvek suit, 9-32.5.1, the sample from stain D on the interior right sleeve of the Tyvek suit, and 16-13.1, the unknown tissue recovered from the Swampscott dumpster.
Yes, I did.
And what work did you do on those 6 samples?
So I performed the second and fourth step of the DNA analysis process, quantification and detection.
And can you tell us how you did the quantification step?
So I took a sample, I was given samples from the extraction step, and then I added a mix of chemicals and went through repeated processes of heating and cooling, which quantified the DNA.
And then the process was then next ready for the third step, is that correct?
Yes, it was ready for amplification.
And who did the amplification step?
I believe that was Madison Frank.
Okay. And after Madison Frank did the amplification step, what did you next do?
I took the products of the amplification step and performed detection.
And did you do the detection on those same 6 items that we just discussed?
Yes.
And how did you do the fourth step, that detection?
I added a mix of chemicals and placed the samples on the detection analyzer.
And then what happened?
And then electropherograms were generated, which were then interpreted by the interpreting analyst.
And where did that data go?
That data was transferred to one of our servers at the State Police Crime Lab.
And did you do the interpreting or did someone else do it?
Someone else performed the interpretation.
Did you do any other work on this case?
Not that I'm aware of, no.
I have no further questions.
Any cross-examination of this witness? The witness may step down. Thank you, sir.
Thank you.
Woohoo! All right. Now we are on witness number 35.
The Commonwealth may call its next witness.
Emily Oliver, please.
Emily Oliver. Let me guess, she's a forensic person. Wow, look at that binder. I wonder how many binders they have full of paper. Woo! All right. She does not have a file folder, so
Given the cause now and hearing will be the truth, the whole truth, and nothing but the truth, so help you God. Thank you.
This way. Let's watch your step.
You may proceed.
Thank you. Good afternoon. Could you please state your name for the court?
My name is Emily Oliver.
And can you tell us your educational background?
Yep. I received a Bachelor's of Science in Forensic Science from the University of New Haven in 2019.
And what is your occupation, ma'am?
I am a Forensic Scientist 2 at the Massachusetts State Police Crime Lab in the DNA unit.
And how long have you been employed there?
Since December 2019.
And can you please tell us your responsibilities?
So at the lab, I perform DNA testing on samples as well as DNA profile interpretation, and I write lab reports.
And what training have you had for this position?
So when I was first hired, I did an in-house training program that lasted over 6 months that involved testing mock casework samples as well as lectures and exams.
And you also have ongoing training?
Yes.
And have you taken competency tests and proficiency tests as well?
Yes.
And have you passed those?
Yes.
Prior to doing any work, how do you prepare your work area?
So first I clean the area and all the tools I'm going to use with bleach, and then I lay down clean bench paper.
And how are you dressed before you work?
I have a lab coat, gloves, a face mask, and a hairnet.
And why do you take those precautions?
To prevent any contamination from myself onto the samples.
I want to direct your attention now to lab case number 23-00596 and asked if you worked on items 7-13.1, sample from area A on the head of the hatchet, item 7-13.2.1, sample from the handle of the hatchet, and item 9-20.1, the sample from stain A on tape with apparent gauze with red-brown stain. Did you do work on those items?
I did.
And what did you do?
So for 7-13.1, the head of the hatchet sample, I did the first 3 steps of our 4-step DNA analysis process, which is extraction, quantification, and amplification. And on the other 2 samples, I did the first 2 steps of our 4-step process in the lab.
Okay, I also want to ask you if you did any sample prep.
So I did, in the sample, it was more so just making sure I pulled the correct tube with the correct case number and item number from our cold storage room. It was previously prepared by someone else.
Thank you. Can you tell us what you did for the first step for the items that you did the first step for?
So extraction is the step where we add chemicals and heat to the sample to release the DNA from inside of the cells in the sample.
Okay, and which items did you do that for?
For 7-13.1, 7-13.2.1, and I believe it was 9-20.1.
Okay, so for all 3 of those you did that?
Yes.
Okay. And did you use equipment for the extraction step?
Yes.
And what equipment did you use?
It's called an Easy One extraction robot.
And what is the biological material now referred to?
A DNA extract.
And what is it now ready for?
The second step, which is quantification.
And how did you perform the quantification?
So I took a small droplet from the extract tubes and put them into a new tube. And then I add different chemicals to that new tube.
All three of those items?
Yes,
Okay. And did you use equipment?
yes, it's called a 7500 real-time PCR instrument.
Okay. And then what happened in the DNA process?
And after that, the samples go to the third step if they have enough DNA, which is amplification.
Okay. And for which items did you do the amplification process? Did you just do that for 7-13.1?
Yes.
Okay. Why did you not do amplification for 9-20.1 and 7-13.2.1?
Just due to scheduling reasons, that was worked best for me.
Okay. So who performed the amplification process for 9-20.1 and 7-13.2.1?
My coworker, Mary Nagel.
Okay. So talking now about what you did do amplification for, item 7-13.1, the sample from area A on the head of the hatchet. Can you tell the jury, how did you perform the amplification process on that item?
Sure. So I add, again, different chemicals to the extract tubes and put it onto an instrument that creates millions of copies of these specific locations on the DNA that we're looking at.
And did you use equipment?
Yes, it's called a thermocycler.
And what happens next in the DNA process?
The last step is the separation and detection step. And that after that last step, we have the DNA profile.
And did you do that fourth step, the detection step?
I did not.
And who did that last step for 7-13.1?
My coworker, Yoli Bregu.
I have no further questions, Your Honor.
I just have a few.
Go right ahead.
Oh, boy.
I didn't hear your name.
Oh, Emily Oliver.
And you have a title?
You can just
said
call
press
me
ignite
Miss
too.
Oliver, if you'd like.
Can you tell the jury a little bit something about your knowledge of inhibitors and how they affect the amplification?
Can't hear you.
Sure. So different materials in the environment can kind of limit the reaction and which could affect the DNA profile that we get in the end.
And can you just tell the jurors what are some of the known inhibitors of DNA amplification?
So dye from blue genes can be an inhibitor. I don't recall any others off the top of my head.
All right. The fact of the matter is that the DNA molecule is a pretty robust molecule, correct?
Correct.
And it's fair to say that things as common as oil and things like grease, they don't inhibit the amplification of the DNA molecule in this testing, do they?
I'm not sure.
What about the quantities that you're dealing with? Is it fair to say that the quantity that you're amplifying can be measured in nanograms?
Yes.
And what's a nanogram?
I believe it is, so it's just a very small number. I believe it's 1 × 10⁻12.
So is it fair to say that it's about a one billionth of a gram?
I'm not sure.
Okay. And what about a picogram? Can you define a picogram for the jurors?
It is a slightly larger number, but still a very small quantity.
Okay. And in the amplification process that you just told the jurors about, what happens is that you separate the alleles from the molecule, the DNA molecule, and then you amplify those alleles, and then they reunite into a molecule and a DNA molecule, and then your machine and apparatus that you use, it's able to process it and give a result, correct?
Correct. And that's mostly referring to the fact that DNA is a double helix kind of looks like a ladder that's twisted, and it becomes single-stranded during part of the process, and then comes back together again.
All right, and finally,
Hitting her with some tough questions.
you're familiar with the term electropharogram, correct?
Yes.
And can you just describe for the jury what an electropharogram is?
So it's just a graphical representation of the DNA profile, kind of like an EKG. There's a bunch of different peaks that correspond to the DNA. That's a printout that we get and that we look at the end of our testing.
So in the State Police Crime Lab, when you look at the electropharogram, does the analyst interpret the peaks to determine the results of the DNA testing, or is it another piece of apparatus that does the analysis of the electropharogram peaks?
So we put our electropharogram through a program called Gene Mapper that does help the interpretation, but a person is still looking over the profile.
All right. So is it fair to say that in the State Police Crime Lab, that the final call, so to speak, the final determination of what is or isn't interpreted as DNA, that's done by the analyst, not by the machine?
Correct.
Thank you, Ms. Oliver.
Okay, thank you.
All right, that is witness number 35. Okay, now we have a whole bunch of names. Who's up next? Are we going to hear from
Calls next witness.
Mary
Come up and call Mary Nagel.
Nagel? All right. All right, Mary Nagel.
Do you solemnly swear or affirm the testimony you'll give in the cause now and hearing will be the truth, the whole truth, and nothing but the truth, so help you God? Thank you. Please sit.
You may proceed.
Thank you very much, Your Honor. Can you state your name for the record and just spell your last name?
Yep. My name is Mary Nagel, Nagle.
And what's your educational background, ma'am?
I have a bachelor's of science in biochemistry, cell and molecular biology from Drake University, as well as a master of science in biomedical forensic sciences from Boston University.
Can you just pull that microphone a little bit closer? Thank you. And what is your occupation?
I am a forensic scientist too in the DNA unit at the Massachusetts State Police Crime Lab.
And how long have you been doing that?
Just over 3 years.
Can you tell us your responsibilities?
Yep, I am capable of performing all of the lab work as well as reporting and interpreting single source profiles and writing up other reports as well.
What training have you had for this position?
I underwent a 6-month training program that included lectures, exams, and about 50 mock samples for lab practice.
And did you pass the exams that you during your training period?
Yes, I did.
And you also have ongoing training?
Yes, I do.
And as part of your ongoing training, do you have to take proficiency tests?
Yes, we take 2 proficiency tests a year.
And have you passed those tests?
Yes, I have.
Directing your attention to lab case number 23-00596. Did you do work on item 7-13.2.1, the sample from the handle of the hatchet, and item 9-20.1, the sample from stain A on tape with apparent gauze with red-brown stains?
Yes, I did.
And what did you do on those items?
I performed the 3rd and 4th step of the DNA analysis, which is the amplification and detection process.
And how did you do the amplification step?
For amplification, I was made aware of which samples to process from Emily Oliver. From there, the samples were set aside in a tube, and I took whatever amount was needed specific to that sample item and added chemicals to that. The samples and the chemicals were then put on an instrument that went through a series of heating and cooling. And this process makes millions of copies of that DNA.
And did you use equipment?
Yes, for that, it is mostly just the heating and cooling instrument that is used, and then the pipettes and tubes and instruments like that as well.
And then you said you did the 4-step detection?
Yes, I did.
And which did you do the detection for both of those items that we previously discussed?
Yes, I did.
Okay. And how did you do that?
So after the 3rd step amplification, the samples are in smaller tubes, and a small portion of that sample is then loaded onto a plate with another set of chemicals. That plate is loaded into an instrument to run, and that instrument then detects the DNA and produces a profile for interpretation.
And did you do the interpretation?
No, I did not.
Who did the interpretation?
That was Saman Salim.
I don't have any further questions, Your Honor.
Any questions of this witness? You may step down. Thank you.
Thank you.
Oh, I'm sorry.
I need to speak up. Ms. Nagel,
Yes.
just have a few questions for you. Once again, there are certain known inhibitors that affect the ability to amplify the DNA molecule, correct?
There are, yes.
But there's not very many
Correct.
because it's a robust molecule, correct?
Right.
One of the inhibitors is, interestingly enough, the blue dye that we see in things like Levi's, correct?
It can be, yes.
Okay. But it's not something like oil or grease or things like that. The molecule doesn't get broken down by oil and grease and things like that, correct?
Correct.
And if bleach is used on an item that contains DNA and the bleach breaks down the molecule, that molecule will not properly amplify using your crystal.
It will still be amplified, but parts of that DNA will probably drop out at the end of it, but there is still usually DNA at the front of that profile.
When you say at the front, you mean that the shorter alleles, after the molecule is broken down, the shorter alleles will amplify, but the longer alleles won't because they were broken down by the bleach.
Correct.
And when you look at the results of their amplification, a trained analyst can see that the longer alleles didn't amplify properly, and they may conclude that something like that particular sample might have been affected by bleach or something like that, correct?
We do not look into what could possibly affect the degradation, but we are capable of seeing the fact that the DNA profile will begin to slowly drop in what is present.
Okay, thank you. One other question. Am I correct in saying that a nanogram is 1/1000th of a gram?
Yes, I believe so.
Okay. Am I correct in saying that a picogram is 1,000th of a nanogram?
A picogram would be 1/1,000,000th.
And when you're using the quantities that you're using to get the results you complete in the lab, we are talking about quantities in nanometers.
Yes.
Tiny, tiny amounts, correct?
Yes.
Thank you. Thank you very much.
Keeps making that point that it's such
Any redirect
so
of this witness?
tiny.
No, Your Honor.
The witness
All
may step
right.
down.
Are we going to now hear from, I'm assuming what's Yuli Break? I got that right? Because he did the 4th step on the head of the handle of the hatchet.
Commonwealth may call its next witness.
The Commonwealth would call Yoli Bregu.
I didn't say his name right, but I was right. Okay, 37. So he did the 4th step on the handle, hatchet, something another.
Do you solemnly swear or affirm the testimony you'll give in the cause now and hearing will be the truth, the whole truth, and nothing but the truth, so help you God? Thank you.
Head of the handle, hatchet.
Counsel,
Oh,
can you
it's a
hold that for
girl.
a second?
I thought it was a boy. Guess you make assumptions with the names. I want to see how this person's name is spelled. Sounds really interesting. And then I can't wait for the end person, the analysis last girl, to come in. She's got a lot to testify to. She's gonna be there for a long time because she did all
You may
the
proceed.
analysis on all of
Thank
these.
you, Your Honor. Good afternoon.
Good afternoon.
Could you please state your name and spell your entire name for the record?
My name is Yoli Bregu, Jolibregu.
And what is your educational background?
I have a Bachelor of Science in Biology from Boston University and a Master of Science in Biomedical Forensic Sciences, also from Boston University.
And what is your occupation, ma'am?
I'm a forensic scientist too with the Massachusetts State Police Crime Laboratory in the DNA unit.
And how long have you been there?
Since June 2019.
Can you tell us your responsibilities as a forensic scientist too?
Sure. I perform forensic DNA analysis on evidence samples submitted to the lab. So my typical workday consists on processing those samples in the lab, doing the data interpretation, making comparisons with the reference samples, writing reports, testifying in court as needed. I also do administrative and technical reviews.
And what training have you had for this position?
So in addition to my education during my master's degree, I followed a forensic biology and forensic DNA analysis track. And then when I got hired, I was trained at the Massachusetts State Police, specifically in the DNA unit in forensic DNA analysis. The training consisted on lectures, reading of relevant literature, laboratory experience with practice samples, data interpretation, written and oral exams, followed by competency tests.
And did you pass those competency tests?
Yes.
And do you have ongoing training as well?
Yes, we have ongoing training. Every year we are required to take at least 8 hours of continuing education, which is offered in forms of seminars, webinar, workshops. We usually take more because there's more offered through the year. We're also required to read at least one scientific journal article relevant to our field.
And do you have to take testing as well?
Yes, we have to take proficiency testing every year. They are taken twice a year.
And have you passed that proficiency testing?
Yes, I have passed the proficiency testing required throughout my employment. The last one is still in the grading process.
And have you testified in court about DNA?
Yes, I have.
Directing your attention to lab number 23-00596, did you do work on item 7-13.1, the sample from area A on the head of the hatchet?
Yes, I did.
And what work did you do on this item?
I performed the fourth step that is used in forensic DNA analysis, which is detection.
And how did you do the detection step of the DNA process?
So I took the samples from the analyst who had done previous work, Emily Oliver. I prepared those samples, mixing them with the required reagents. Then I loaded them in our instrument that does the detection, known as genetic analyzer, and I started the run. And that's where my role ended. After the run is done, the data is saved automatically in the computer and can be imported by the interpreting analyst for data interpretation.
Did you do the interpretation here?
No, I did not.
Who did the interpretation?
Salman Salim.
I have no further questions, Your Honor.
Any cross-examination?
I'm just going to pause there. So out of all of them, right? Out of all of the analysts, right? It's like, which one's your favorite? I really liked her because she really, so I'm a sciencey, geeky nerd person. I really liked listening to that. And when she was talking about the reagents, those are the chemicals that they use because they said, always use chemicals. Well, some people like a little bit more technical. So she used the reagents. I really like that. So just wanted to say that there. All right, let's see if Mr. Tipton has any questions for her.
This witness. Go right ahead.
Is he going to ask the same questions? She seems to know her stuff. She might be the one to ask.
Couple of questions. You're familiar with the term in the context of DNA testing, allele, correct?
Correct, yes.
And the allele is, why don't you just tell me?
The allele is a variant of a DNA fragment that is amplified during our third step. So it's a person has two alleles, one they inherit from the mother and one from the father.
Okay. And the allele then is the section of the DNA molecule that is then looked at once you've separated the molecule, and it's the part of the molecule that then gets amplified, correct?
Yes.
And so if you have an allele that repeats at a certain locus on the DNA molecule, then you may be a particular type at that particular location on the molecule, depending on how many repeating alleles you have.
It's how many repeats are within the allele. So the allele itself is the entire fragment, and within that alleles, there are we call them short tandem repeats. There are repeats, they repeat themselves different numbers for different individuals, and that's what it's assigned to an allele. How many times the fragment repeats itself within the allele.
And people can have the same repeats of alleles in the molecule, but generally speaking, they don't have the same number of repeating alleles across the molecule, various different loci.
Yes, correct.
And the loci is just plural for locus, right?
Yes, the loci is the location of that particular allele in the DNA molecule.
In the testing that was done in this case, you're using 26 different locations plus the sex area.
Correct, yes.
That's a mellogen.
Correct, yes.
Thank you.
Any redirect of this witness? No. The witness
That
may
was
step
a
down.
very beautiful question,
Thank you,
Mr.
Your Honor.
Tipton.
Thank you.
People got a little science lesson. All right. We are on witness number 38. And the court is at approximately 12:38. So, are they going to go to lunch? Oh, let's see what she has to say.
You, the judge.
Oh,
Days
maybe
ago,
she's
I
got.
told you, and I think I read you another stipulation, but I'll remind you, a stipulation is just an agreement between the parties, and I'm going to read you now an agreement that the parties have made.
Okay, so stipulation.
This is a stipulation regarding DNA samples of Anna Walsh and Brian Walsh. Reads as follows. On May 9, 2018, FBI Special Agent Tyler Delorme obtained a DNA sample from Brian Walsh.
The
Special
FBI.
Agent Delorme brought the sample to the FBI headquarters in Boston, where it was placed into evidence storage. Also on May the 9th, 2018, FBI Special Agent Matthew Riportella obtained a DNA sample from Anna Walsh.
Oh.
Special Agent Riportella brought the sample to the FBI headquarters in Boston, where it was also placed into evidence storage.
Huh.
On January 10th, 2023, Special Agent Kristen Koch took the sample of Anna Walsh and the sample of Brian Walsh from evidence storage at the FBI headquarters in Boston and brought both samples to the Massachusetts State Police Crime Lab. The samples did not leave the custody of the FBI until Special Agent Koch submitted them to the Massachusetts State Police Crime Lab for testing.
Oh boy.
That's exhibit.
Let's talk about that. Okay. So, in May of 2018, this is when his art fraud case came into play. Now, I can see the FBI taking his DNA because he then is like in their custody and control. It's something like that, right? But her DNA That is interesting. I will have to, that is interesting. Why did they take her DNA? Huh. I don't know. Let's see if we, well, you know what? Here we are. Real time Google. Let's see what Google has to say. Why did FBI take Anna Walsh's DNA in 2018 during Ryan's fraud case? Let's see. Oh, okay. So it says the specific reason for her DNA was collected was related to the allegations that Brian may have used her eBay account to sell the fake Andy Warhol paintings. Oh. Huh. All right. Well, there we go. Real time answers. All right, let's continue. They're going to go to lunch soon.
E for identification.
Well, that answers that.
Commonwealth may call its next witness.
The Commonwealth
Because-
will call Karen Jacobson.
Because she might have been implicated. Okay, witness number 38, Karen. Somebody another. Karen Jacobson. Oh, she's got her thick binder.
Do you solemnly swear or affirm the testimony you'll give in the cause now and hearing should be the truth, the whole truth, and nothing but the truth, so help you God? Thank you.
Right this way.
All right, what did Karen do? Do we have more stuff that they analyzed? I thought we were done.
You may proceed.
She's got a file
Thank
folder.
you very much, Your Honor. Good afternoon.
Good afternoon.
Could you please give us your name and spell your name for the record?
My name is Karen Jacobson. That's KarinJacobsen.
And if I could just ask you to pull that microphone a little closer to you. Thank you.
Sure.
What is your occupation, ma'am?
I'm currently employed as a forensic scientist 2 within the DNA unit at the Massachusetts State Police Crime Laboratory
And how long have you been there?
since August of 2021.
And what are your duties?
I'm responsible for receiving items of evidence associated with known individuals, taking them through the 4-step DNA process, and making those DNA profiles available for use in comparisons.
And what is your educational background?
I have both a Bachelor's of Science in Forensic Science and a Master's of Science in Forensic Science from Bowling Green State University.
Bowling, Kentucky.
And do you have specialized training to be a DNA analyst?
Yes, I do.
And can you tell us about that training?
The training program within the DNA unit is at least 6 months long. It consists of lectures covering all of our protocols, literature readings covering current and historic methodologies, laboratory processing of at least 50 mock samples, and all of this is wrapped up with a competency test.
And did you pass your competency test?
Yes, I did.
And have you also had ongoing training?
Yes, I have.
And as part of your ongoing training, do you have proficiency testing?
Yes, I do.
And have you passed your proficiency training?
Yes, I have.
I want to ask you about lab number 23-00596. Did you do work on this case?
Yes, I did.
What work did you do on this case?
The DNA testing process is 4 steps long. I performed the 2nd step and the 4th step for 2 items.
And what were those items?
These were items 13-1.1 and 13-2.1.
And what were those?
These were the DNA saliva collection kits for Anna Walsh and Brian Walsh, respectively.
Okay. So how did you do the step 2? That be the quantification step?
Yes.
Okay. Tell us about that, please.
In the 2nd step, quantification, I received a portion of the extract created in the 1st step.
I applied a set of chemicals to it and put it on an instrument that goes through cycles of heating, cooling, and this produces an estimate of the amount of human and male DNA present within each sample.
And did you use equipment?
Yes, I did.
Okay. And what was that?
This is a 7500 real-time PCR machine.
And who had done the previous step?
The previous step was performed by Katerina Stashin.
Okay. And why didn't you do that step?
We batch our samples in the laboratory processing for the sake of efficiency.
Okay. And then when did you become involved again in this process for these samples?
I then performed the fourth step, separation and detection.
Okay. And can you please tell this jury how you did that fourth step?
So the same person, Katerina Stashin, performed the third step. I received the product produced in the third step from her. I applied another set of chemicals to it and put it on an instrument that separates each of the copies of the DNA by size. And then with the help of some special software, it turns it into a visual representation of the DNA profile.
And did you use any equipment?
Yes, I did.
And what equipment did you use?
This was a 3500 genetic analyzer.
Okay. And then what is the data next ready for in the process?
After the data is reviewed, it's ready for use in comparisons.
Okay. And who did the interpretation and comparison here?
In this case, it was Katerina Stashin as well.
And did you do any other work on these samples?
No, I did not.
I have no further questions.
Cross-exam? The witness may step down.
Thank you.
All right. So she processed the saliva from the FBI. She did steps 2 and 4.
Commonwealth
Katerina
may call its
did
next
1,
witness.
3, and
The
the.
Commonwealth would call Katerina Stashin.
Okay. Witness 39. Katerina. Okay, so she did the saliva samples from the FBI. She did steps 1
Do you solemnly swear or
and
affirm the testimony
3,
you'll give in the cause
and
now
then
and
she
hearing
did the
nothing but the truth, so help you God?
comparison analysis. So she's going to be a little bit longer because she has like the end results of everything. Most of these people are girls.
Thank you very much, Your Honor. Good afternoon. Can you please state your name and spell your whole name for the record?
Good afternoon. My name is Katerina Stashin. First name is spelled Katarina. Last name is spelled Stasshyn.
And what is your current occupation, ma'am?
I'm currently a criminalist in the biology section at the Washoe County Sheriff's Office Crime Lab located in Reno, Nevada.
And can you please
She doesn't
detail
work
your
in
work history
MSP
as it
anymore.
applies to forensic sciences?
I was previously a forensic scientist in the DNA unit at the Massachusetts State Police Crime Laboratory for about 3.5 years, from 2021 to earlier this year, 2025, when I got the opportunity to move back to Nevada to be closer to family and pursue a job still as a DNA analyst.
And what is your educational background?
I have a bachelor's and master's degree in biotechnology from the University of Nevada, Reno.
And do you have any specialized training to be a DNA analyst?
Yes, I received training at both the Massachusetts State Police Crime Lab and at my current job in Washoe County. In Massachusetts, I completed an approximately 1-year-long training program that consisted of reading scientific protocols, scientific literature, textbooks, all on the topics of forensic DNA analysis. I also took written exams and oral exams on those topics. I also practiced sets in the lab, both for laboratory processing and for DNA interpretation. Put together practice case files, wrote reports, did comparisons, and all of that training completed with a competency test, of which I passed. And then at Washoe County, I completed an approximately 6-month-long training course due to my experience, where I also did review of scientific literature, textbooks, protocols, practice sets, writing practice reports, performing comparisons or interpretation, etc. And that also completed with a competency test of which I passed.
And have you also had proficiency testing?
Yes, I have also completed proficiency testing.
And have you passed those tests as well?
Yes, I have passed all competency and proficiency testing that I've taken so far.
And have you testified in Massachusetts about DNA?
Yes, I have.
So what is a single source DNA profile?
A single source DNA profile is a profile that originates from one person or one contributor. And you can identify this in a profile. Just by looking at the profile, you can see how many people are in there.
And what is a known standard?
A known standard is a sample that's taken directly from an individual. For example, a sample from somebody's mouth, a saliva swab, or a blood sample. It's taken directly from a person to be used for comparison purposes.
And how is a known standard developed into a DNA profile?
A known standard will go through the four-step laboratory process to result in a DNA profile that can then be used for comparisons.
And the four-step process is what?
The four-step laboratory process for DNA analysis is extraction, quantification, amplification, and detection.
So prior to doing any work, how do you prepare your work area?
So when you first get into the lab to do any of those steps, you're going to clean your lab bench. So that involves using a bleach spray to wipe down your area. You're going to clean all of the equipment, utensils, your pens, pipettes, any equipment that you're using. And then me as a person or any lab analyst, you'll be wearing a lab coat, a face mask, a hairnet. You're in full PPE, wearing gloves, changing gloves frequently.
Why do you clean your area and wear PPE?
We do this just to keep everything perfectly clean, or the cleanest that we can, just to help the integrity of the evidence and help produce the best possible profile.
Now I'm going to direct your attention to Massachusetts Case Lab 23-00596. Did you do work on this case?
Yes, I did.
And what did you do on the known standards in this case?
I performed the sample prep, the extraction, and the amplification on the known standards in this case.
And did you also do interpretation on this case?
Yes, I also reviewed the profiles that were obtained from that process and made sure they were able to be used for comparison purposes.
So I want to ask you about each of the things that you just told us about. So were you working on 13-1 and 13-2? Is that correct?
Correct.
And what were those?
Those were the known saliva standards for both Anna Walsh and Brian Walsh.
And so what was 13-1?
13-1 was the known saliva standard for Anna Walsh.
And what was 13-2?
13-2 was the known saliva standard for Brian Walsh.
And how did you do the sample prep for 13-1?
So both of these samples were known saliva samples, so a swab from the mouth, and I just cut up the swab and placed the cotton part of the swab into a tube, a labeled tube with those item numbers that I said.
Okay, so that's what you did for 13-1, and then did you give it another number?
So once I cut that swab off, it then became 13-1.1.
And so did you do the same thing for 13-2?
Yes, so then the cotton that got cut off of the whole swab became 13-2.1.
And then after you did that sample prep, what did you next do?
Then those samples went through the extraction step of the process.
And how did you do the extraction step?
So extraction consists of adding chemicals to the samples and going through heat variation to help break open the cells present in the sample and release the DNA that is present. And then that DNA is isolated.
And did you use equipment for that?
Yes.
What equipment?
An incubator was used for that heat variation that I mentioned, and an easy one robot was used for the extraction part, meaning that after it sat in the incubator with the chemicals to break open the cells, a robot is used to isolate that DNA from the rest of the cellular debris left behind.
So you told us about sample prep and step 1. Did you do the next step, step 2?
No.
Why not?
Just based on scheduling at the lab, who was assigned to do what step that day.
And who did that next step?
The second step of quantification was performed by Karen Jacobson.
Were you qualified to do all of the steps?
Yes.
And so what did you next do?
I next did the amplification step, that third step I mentioned.
And how did you perform the amplification step?
So amplification is the third step, and this is when we make millions and millions of copies of certain fragments of DNA. So this is done by, again, adding certain chemicals and going through heat cycles to make these many millions of copies of DNA. Because we're usually working with a smaller amount of DNA, you need to make a lot of copies to make it visual and able to be detected.
Did you use equipment?
Yes.
What equipment?
We use what is called a thermal cycler, and this is the instrument that performs those heat cycles needed to make the copies.
So did you do the next step, the fourth step?
No.
Who did that?
Karen Jacobson also performed the fourth step of detection.
So then what did you do next?
So after that full laboratory process was completed, I looked at the data that was produced from those 4 steps, and both items resulted with DNA profiles that I reviewed and made sure were acceptable to be used for DNA comparisons.
And could you visualize a single source?
Yes.
And did you generate a DNA profile for Anna Walshe?
Yes.
And did you generate a DNA profile for Brian Walshe?
Yes.
And were profile sources developed that were appropriate for comparison?
Yes, the profiles developed were acceptable to be used for comparisons.
And they were both single sources?
Yes.
What does appropriate for comparison mean?
When I'm saying appropriate for comparison, I mean that the profile is full, as in data is obtained at every location that was tested, and that it is single source.
And what do you do with the profiles?
That profile is then made into a text file that is made available for the analyst that's going to be doing the comparisons.
And were the profiles assigned a number to identify them?
Yes, so the numbers for the profiles are the same as their item number. So the text file profile for Anna Walshe also had the number of 13-1.1, and the text file profile for Brian Walshe was 13-2.1.
If I might have 1 second, Your Honor.
You may.
Nothing further.
Cross-exam. The witness may step down. Thank you.
Thank you all.
Okay. Oh boy, that was a lot, right? That was the like very heavy science-y thing, and they have to go through each process, etc. Okay, so they're going to take their lunch break right now. Okay, so I'm going to go ahead and end this one because I'm trying to do quick, strippy ones. So we're going to end this one. Science can be a lot. So, you know, it is what it is. But if you're a science tech, a science nerd, that you want to know a little bit more about this, this was very telling. It kind of was quite repetitive, but they have to go through the repetitive motions because this is evidence, and they have to technically say, what did you do to get this evidence? And so just like they have a 4-step process and not everybody's doing the process is all in one for all of these things. Again, I understand, but boy, it's a lot, right? So that's where we're at. So we're going to stop this one. We're going to pick it back up after lunch and see where we get. I think we have one more forensic scientist expert to come in because she did all of the analysis that'll tell us whose DNA is on what, right? Because that's the end result. That's what we want. Whose DNA is where. So we'll see you again in just a little bit. Make sure you follow and like and subscribe so you can hear more content. See you in a bit. Thanks for watching Legalese 123. If you like what you saw, please like and subscribe for more content.